Biology with Dr. E.
Next, we look at osmosis. Osmosis is the movement of water through selectively permeable membranes. It's important in determining the distribution of water across various fluid-filled compartments in the body. Water crosses the plasma membrane through two main mechanisms. One is through simple diffusion across the lipid bilayer. Please note that although water is polar, it's able to cross the lipid bilayer. The second mechanism is through special channels called aquaporins. Osmosis is important in balancing the solute concentration across membranes. It occurs whenever the solute concentration differs on two sides of a membrane. This figure illustrates osmosis. Notice that water molecules can cross the plasma membrane through a specific channel called aquaporins or directly through the lipid bilayer. Next, we review osmolarity. Osmolarity is the total concentration of all solute particles in a solution. Typically, water concentration varies with the number of solute particles in that solution. So when solute concentration goes up, water concentration typically goes down and vice versa. Water would typically move from a region of low solute to a region of high solute concentration. That is to say, water moves from a region of high water concentration to a region of low water concentration. Osmosis occurs in an attempt to balance the osmolarity on either sides of a membrane. When solutions of different osmolarities are separated by a membrane permeable to all molecules, diffusion of solutes and osmosis occurs until equilibrium is reached. At equilibrium, there is an equal concentration of solutes and water across the membrane. This figure shows a U-shaped glass tubing containing two different solutions. The left compartment of the tube contains a solution with lower osmolarity or lower solute concentration. The right compartment of the tube contains a sugar solution with higher osmolarity. The two solutions are separated by a semi-permeable membrane that's freely permeable to both solutes and water. The solutes and water move down their concentration gradient in opposite directions. At equilibrium, the resulting solutions on each side of the membrane have the same osmolarity, and the volume of the solution in each compartment is unchanged. When solutions of different osmolarities are separated by a membrane that is permeable only to water, only osmosis will occur until equilibrium is reached. Water will have net movement across the membrane until osmolarity is balanced on both sides. This results in volume changes on both sides of the membrane. The volume of the side with lower solute concentration will decrease, while the side with higher solute concentration will increase. This figure here illustrates a U-shaped glass tube containing two solutions. The left compartment of the tube contains a solution with lower osmolarity or lower solute concentration. The right compartment of the tube contains a sugar solution with higher osmolarity. The two solutions are separated by a selectively permeable membrane. The membrane is only permeable to water but impermeable to solutes. So water but not solutes can move through the membrane. Water moves by osmosis from an area of lower solute concentration to an area of higher solute concentration. As a result, both sides have the same osmolarity but different volumes at equilibrium. Next, we look at tonicity. Tonicity is the ability of a solution to change the shape of a cell or the plasma membrane tension of a cell by altering its internal water volume. We would review three types of solutions: isotonic solutions, hypertonic solutions, and hypotonic solutions. Cells placed in any of these solutions can alter their shape based on the concentration of the solution. An isotonic solution has the same osmolarity as inside the cell. So the cell retains its normal size and shape with an unchanged volume. Water moves into and out of the cell with no net gain or loss of water. A hypertonic solution has a higher osmolarity than inside the cell, so water flows out of the cell, resulting in the cell shrinking. There is a net loss of water from the cell. Shrinking is also referred to as crenation. A hypotonic solution has a lower osmolarity than inside the cell, so water flows into the cell, resulting in cell swelling. There is a net entry of water into the cell. This can lead to the cell bursting, also referred to as lysing.